In a groundbreaking feat, Colossal Biosciences, a Texas-based company, has successfully cloned dire-wolf hybrid pups. This marks a pivotal moment in the realm of de-extinction, raising both excitement and ethical concerns. The dire wolf, a formidable predator that roamed North America during the late Pleistocene epoch, vanished approximately 13,000 years ago. Thanks to cutting-edge genetic technology, a semblance of this majestic creature has been brought back to life. Colossal Biosciences aims to use this technology to not only revive extinct species but also to contribute to the conservation of endangered ones. The company's efforts have garnered attention worldwide, sparking debates about the ethics and potential consequences of such scientific advancements. The birth of the dire wolf pups is just the beginning of Colossal's ambitious ventures into de-extinction. The company's work has far-reaching implications for our understanding of evolution, genetics, and the delicate balance of ecosystems. As we delve deeper into the world of de-extinction, we must tread carefully, considering the ethical ramifications and potential ecological impacts of our actions. The successful cloning of dire wolf hybrid pups is a testament to human ingenuity, but it also serves as a stark reminder of our responsibility to protect the natural world. The journey from fossil DNA to living pups involved a complex series of scientific procedures. Firstly, scientists extracted DNA from well-preserved dire wolf remains, including a 13,000-year-old tooth and a 72,000-year-old skull. This genetic blueprint was then compared to the genomes of modern canid species, primarily the grey wolf, to identify the key genetic variants responsible for the dire wolf's distinctive traits. These traits, which set the dire wolf apart from its modern relatives, included a larger size, a wider head, thick fur, and a significantly stronger bite force. Once these genetic markers were identified, scientists at Colossal Biosciences employed advanced gene editing techniques to modify grey wolf cells. They made a total of 20 edits across 14 genes, essentially rewriting the genetic code to incorporate dire wolf characteristics. These modified cells were then used to create cloned embryos through a process called somatic cell nuclear transfer, or SCN. This technique involves removing the nucleus from a donor cell, in this case, the modified grey wolf cell, and inserting it into an egg cell that has had its own nucleus removed. The resulting embryo is then implanted into a surrogate mother, which carries the pregnancy to term. For the dire wolf project, Colossal Biosciences used large mixed-breed domestic dogs as surrogates. The first dire wolf hybrid pups were born in October 2024, followed by a female in January 2025. These pups carry dire wolf genes but are predominantly grey wolf. This has sparked a debate about the true definition of de-extinction. Some argue hybrids can't be true dire wolves, while others see it as a step toward restoring biodiversity. The pups live at a secure facility monitored by Colossal Biosciences. The de-extinction of the dire wolf has ignited ethical debates surrounding the use of such powerful technology. Critics question the allocation of resources, arguing funds could be better directed toward conserving existing endangered species. Concerns include the welfare of surrogate animals and the ecological impacts of introducing a genetically modified predator. Colossal Biosciences argues its work has significant conservation implications. Technologies like gene editing can bolster the genetic diversity of endangered species. The company has produced cloned red wolves, a critically endangered species. The debate is complex and multifaceted, with no easy answers. It is crucial to proceed with caution, engaging in open discussions about the implications. The future of de-extinction remains uncertain, but the successful cloning of dire wolf hybrid pups marks a significant milestone.